Thank you for the introduction. This is joint work together with two biomedical research centers in Germany, which mainly provided us with the necessary insights into the biomedical field. And my talk is split into two different parts. The first part is mainly about demonstrating the privacy risk inherent to these two types of biomedical data, namely the genome and DNA methylation. The second part of my talk then focuses on securely classifying DNA methylation samples at a third-party laboratory. For example, using random forests, as we did in our protocol that I will present. Okay, let me briefly introduce you some of the background needed for this talk. Okay, the genome. The genome of a human consists of 4 different nucleotides, namely A, T, G, and C. And these nucleotides form a long sequence called DNA sequence. And if you compare the DNA sequence of any person with the DNA sequence of any other individual, then you will observe that these two DNA sequences are nearly identical. But there are still some positions in the human genome which differ between different individuals. And these are the interesting positions in the genome, and they are so-called SNPs, or single nucleotide polymorphisms. There are around 150 million of these SNPs in the human genome, and we can characterize mathematically each of these positions by one value. It is either 1, 0, or 2. Next, I will introduce you to the background on DNA methylation. DNA methylation is actually a certain molecule which can occur in the DNA, and it can only occur at certain positions in the human DNA. Namely, it can only occur at those positions where a C is followed by a G in the DNA sequence. This makes around 30 million of these positions where DNA methylation can possibly occur. And whether it actually is methylated or not, such a position, this is highly influenced by the environment, by external factors, or the genome itself. And as this DNA methylation can vary between different cells, we actually measure it on a bunch of cells, and then each methylation at a certain position is characterized by a value between 0 and 1, which denotes the fraction of cells which have been methylated at this position. Let me move to the first part of my talk. So, as this recent article by The Guardian has pointed out, there's a huge business behind medical data and selling this medical data. And those companies and data miners who do this kind of business, they're not only collecting the data, but they are also cross-referencing it across different sources. And as this graph shows, there are a lot of databases out there in the internet where also methylation data and also the genome is publicly available. And this graph shows the amount of data over the last recent years that is available in one of these databases, in the geodatabase. And it grows somewhat exponentially. So there's a lot of data out there available which can be used by those companies and which can be cross-referenced. And so in this talk and in the paper, we assume that the adversary has access to 2 types of biomedical data, namely the genomes of a large set of individuals, and he also has access to a database of his victims where only the DNA methylation profile is known. Now, the adversary tries to link the DNA methylation profiles to the genomes in the other database. But the adversary does not necessarily know that the correct or corresponding genome is actually present in the data. So it might be as well that the output of his attack is the corresponding genome is not present at all. Okay, if we formalize this kind of attack, then we can say that the adversary aims, given a DNA methylation profile, to find the most probable genome in the second database. And if you look at a specific position of DNA methylation and a specific position of SNPs, this graph denotes is a histogram on the methylation levels at a certain position, and I colored the histogram depending on the SNP value at another position. And you can see that these histograms are quite distinct. So given the methylation level at a certain position, in this graph we are actually able to predict the SNP value at another position by just looking to which of these colors here it would belong. And this is also the intuition we will use in this kind of attack. And we make it a bit simpler by pre-selecting independent pairs of DNA methylation and SNPs where there is a high correlation between these two types of data at these positions. So we specifically select those pairs where the genome highly influences the methylation. And when we have those independent SNPs for each of the pairs, we can characterize the adversary's goal by this probability. And actually, if we then rewrite it as this formula, then we can see that it only consists of two probabilities. The latter one can be learned from population statistics, and the first one, the probability of a certain methylation level given a certain SNP value at another position. It can be easily learned from a training data set. And this corresponds to exactly this graph I showed to you. This denotes the histograms for the methylation levels given a SNP value. And as you can see here, this distribution, it looks somewhat like a normal distribution, and that's also the assumption we make in our paper. And we learn these normal distributions from a training data set, and then we can use the probability on top of the slide to estimate the probability, the most probable genome given a methylation level. Now, since we have a lot of these different pairs, the adversary somehow has to map them to one score for each pair of methylation profile and genome in his two databases. And we do this by taking the average over these probabilities for a given methylation profile M and a genome G. And now the adversary's task is to finally find an assignment between those two databases which maximizes this match score. And if we have multiple methylation profiles in multiple genomes, this can be done, for example, by a best vertex matching on a weighted bipartite graph. So maybe you remember I told you that the corresponding genome does not have to be present in the genome database. So we still need another additional step, and this is a statistical validation, and we apply a statistical z-test there to find out whether the assignment we found is really the correct genome or whether the correct genome is simply not present. We carried out this attack on a data set consisting of 75 individuals, and in those 75 individuals, there are also 21 mother-child pairs, so we also have some samples of really closely related individuals in there, which makes it harder for the attacker. And we have the full genome and also all methylation sites available, and after filtering for those positions that are highly correlated, we end up with around 300 of these pairs. Now let's have a look at the results we get. So we varied the number of victims in the right-hand side database and also the number of genomes in the genome database. This graph now denotes the outcome of the attack for 75 genomes and only one victim. And on the x-axis, we varied the threshold for the statistical validation, but the red curve here shows the accuracy before the statistical validation. What we can see here is that before the statistical validation, our attack already performs really good. We have a matching accuracy of more than 97%. And if you then, on top of that, apply this statistical validation, then we can find a certain range of thresholds for which we even end up with no false positives at all and a true positive rate of 1. Now, we also increased the number of methylation profiles, so the number of victims the adversary has. And then by doing this, we also introduce additional constraints in this bipartite graph matching, and thus the matching accuracy before the statistical validation already goes up, and now we have a matching accuracy of 100%, but still the same threshold that applied before can serve again for a true positive rate of 1 and a false positive rate of 0. Okay, I have to admit that in these two examples, the correct corresponding genome was always present in the database. So we also varied the presence probability of the corresponding genome, and we did this here on the x-axis, and the interesting case is at 0, when the corresponding genome is simply not present in the genome database. And then we see that actually we have a quite high chance to match the methylation profile of the victim actually to the genome of a relative, so of the corresponding mother or child. But still, as the blue line on top shows, that our statistical test, our statistical validation is still able to detect those cases and reject those cases where we match to the relative instead of the real individual. Okay, as I said, we also vary the number of genomes in the genome database. And this is done here by adding 2,500 more genomes successively to the genome database. And the interesting thing here is that the matching accuracy remains stable the whole time. That means we do not introduce any genome that is more close to the actual genome so we do not introduce any new genome that increases the match score. Okay, so in this first part of my talk, I have shown you that we are able to provide an attack against these two types of data, which provides already a fairly good matching accuracy without any statistical validation. Using the statistical validation, we are also able to remove all false positives. The attack is really robust, and also what I haven't shown you, but what I want to mention is that actually 30 of those methylation positions are already enough to carry out this attack with such a high success. Okay, this was then the motivation for the defense part. We looked at applications of DNA methylation in practice, and there is, for example, a random forest classifier that is used in practice. It's called MethPet. It is trained on brain tumor cases and is able to distinguish between different types of brain tumors based on DNA methylation profiles. It uses 1,000 trees, all look very similar to the run on the right-hand side. So you always compare the methylation value at one position with a certain threshold, and depending on that, you follow a different branch in the tree until you get to a class label. Now, usually when such a classification happens in practice, this is not done at the hospital where the sample was taken, but it's processed in an external laboratory. And this process usually takes more than 1 day until you get the result back. And we wanted to secure this process by cryptographically means, and this should not only protect the most important part, the patient's data, but also the business model of such a company which provides these classification tasks. And the business model of those companies often is the algorithm itself. They do not want to reveal their classification algorithm, so how it works. And we built this secure classifier on top of the work by Boast et al. From NDSS 2015, and their basic idea was that a decision tree like the one on the right-hand side can be written easily as a polynomial. And then using several building blocks, for example, 3 crypto systems, a comparison protocol, a protocol to change between these encryption schemes, and an Argmax protocol, Boast et al. Provide a protocol for a single decision tree. What we now did, we introduced another additional building block to change the protocol, to change the ownership of an encryption. And based on this, we provide a protocol that is able to securely classify a whole random forest. And we also provide 2 different choices in this protocol. So actually the first choice is either our protocol that can return each tree's class label individually, or it returns only the plurality vote over all the trees. So you have a choice between giving out more information about your trees, but giving also the doctor the ability to have more precise diagnosis. But if you do not want that, you can also return the plurality vote instead. Okay, let's have a look at the performance of our protocol. So in this diagram, you can see the red line, which is the accuracy when we vary the number of trees used in the random forest classifier. I want to stress that the accuracy is the same for running the random forest classification encrypted, so using a protocol, or just normally. So we do not introduce anything which provides a loss of accuracy. It will be the same. And we can see that actually at 100 trees, the accuracy is almost as good as with 1,000 trees. So we could also use 100 trees instead of those initially proposed 1,000 ones. Then there are two more lines which show the total time which our protocol takes when running it over a network. And one line shows if we return all votes individually, and the other one is the protocol with the plurality vote. And what we can see here is that there's almost no difference between these two choices, so there's no disadvantage in choosing one over the other. So if we evaluate this MethPad classifier using 100 trees, our protocol will take less than 2 hours. And if we use all 1,000 trees, it will take less than 12 hours. And we think that it's still practical because in these scenarios, usually you send the data to the external company and the company will return the result the next day anyway. So now we are even faster if it only takes 2 hours instead of 1 day. So to conclude, I hope that I have shown you in the first part that there's a huge potential privacy threat when different types of biomedical data can be combined. And our attack that I presented here is really robust against adding more genomes to this genome database, and it provides a really good accuracy. And in the second part, I presented you very briefly a provably secure protocol which enables the secure classification of, for example, in this case, brain tumor cases in an external facility. So I thank you for your attention, and I'm happy to answer any questions.
Questions?
Have you explored potential for applying a statistical attack against the secure classification mechanism that is stealing the model via the decisions returned by the sampler, by the classifier?
Can you repeat?
Is it possible to steal the decision trees based on the outputs that they produce?
Okay. I think it is quite hard. I would not say it's impossible. So we assume an honest but curious model in our protocol. And I think if you assume a malicious adversary, it's definitely possible to say.
An honest but curious adversary will examine the outputs and may try to reconstruct the procedures that went into producing these outputs.
So if all of the individual tree outputs are given to the client, then I think the adversary can learn something about the trees, but it of course has to run multiple classifications then. If only the plurality vote is given, then it becomes really hard. Yeah. And I don't think it's possible then.
I guess we'll have to wait for the next year.
Any more questions? So I had one really quick. That was a really nice talk. Thanks for giving that, and it's great work. So when you did the statistical validation, you showed that there exists a threshold where you don't get any false positives, but the attacker doesn't necessarily know what that threshold is in advance, correct?
Right, so what we have seen in one of the graphs I also showed here, but I didn't explain it, if you increase the number of genomes in the genome database, this threshold converges to one value. So I think if the number of genomes the attacker uses is really high, he can guess a really good value for this threshold.
Okay, so you can kind of use other data and get a good threshold to apply to future cases, and your suspicion is that that'll give you pretty good false positive rates still.
Yeah, exactly.
Okay, cool. All right, let's thank the speaker again.